Hi, everyone. I'm Bina Shah. Welcome to our family-friendly introduction to all the research that is happening at Project 8P Foundation. And I'm really excited that we have members of the Patient Leadership Board on this call. In particular, I wanted to introduce you all to Faye Minty, who will be presenting a lot of this material to you and has worked so hard as a volunteer in service to all the families here to be able to make a lot of the research that's very technical and scientific digestible to each of you as caregivers. They're really trying to understand what the foundation's doing to help all the 8P heroes. And Faye, we're so lucky to have her because she is a scientist and actually has worked in the lab on some of the basic science that we're actually funding. She also is mom and caregiver to an 8P hero and is based in the UK. And so I'm really, really excited to have her here. And I know that the entire Patient Leadership Board is so happy for all of the hard work that Faye has put in.
Thanks, Nina. Shall I take you to the next slide? There we go.
So today, Faye will take you through much of the deck. We do have some guests that might join us to present some of this to you. I'll go through the vision mission of Project 8P, and then we'll head into part one, which is really understanding the 8P biology. The second part will be understanding Dr. Wendy Chung's research results that we've shared in the past from her publication and from some of her clinical observations of 8P heroes and a lot of the parent reported information that each of you have shared and contributed to that study. And then the third part will just really talk about a lot of the research strategies and approaches for us to try to find treatment and better understand this disorder so that we can help inform and manage the best care that we can for our 8P heroes. And so if we just take a step back for Project 8P Foundation, the last year, the board of directors and I have worked on a 5-year strategic plan at a very high level. And we kept in mind a vision of what we really wanted the world to look like, which is to cure genetic disorders with novel technologies that are accessible to all. And with that, more specifically about the mission for why we're all here and working so diligently is to empower a unified community for chromosome 8P heroes for a meaningful life today while accelerating treatments for tomorrow. And how do we do that? So we came up with 4 priority goals. And a lot of the underlying components of this is really about thinking out of the box and being innovative. And so first and foremost, we're talking about empowering the community, and you'll see that in the 4 quadrants. And that's the community of caregivers and 8P heroes and all of the other individuals that are here to help volunteer their time, dedicate their expertise towards our mission. Accelerating research for 8P related disorders is going to be most of the talk today. The third, and in no order of importance here, is really to deliver a high quality of standard of care. And some of that work is being done through our multidisciplinary clinic, and we'll be expanding that model so that we can hopefully reach many of you all around the world. And then we also believe it's really important that we can't do any of this without partnering with the global difference makers. So if you're here with us, you are making a difference on this planet, and that partnership is really important to us. And we've done a lot of wonderful things, and many of you may know about them, but we've identified over 500 8P heroes. And that's just such an achievement because I can't imagine many years ago not knowing who was out there that had a very similar diagnosis to us. And with that, we launched the 8P registry and biobank. And that's really, the data platform, biospecimens for researchers and our clinicians to help understand this for us. We declared August 8th, which is coming up next week, as Chromosome 8P Day. And hopefully by the time that you see this recording, we can have a lot of really exciting things to announce to you. We did a Moving Mountains conference last year, which was wonderful. And we did it with other chromosome disorders in partnership to really scale this. we published a research study with Dr. Wendy Chung, and as I mentioned a few minutes ago, we launched our first neurogenetics multidisciplinary clinic to also contribute to the data in the registry. And I'd love to pass it over to Faye for a lot more of the science piece of this, and hopefully it's digestible. And we're very happy to have a Q&A session after you've all been able to watch this recording.
Absolutely. Thanks so much, Bina. That's really kind of you. And thanks for joining, and anyone who's going to watch this in the future. So hello, everyone. Let's just begin part one, understanding 8p biology. So when I was thinking about how to set this talk up, I thought we should start with something which is really well established from a source readily available to anyone who might be listening or watching this discussion, and that's Google. That's where I start everything. So Google tells us in the humans, each cell normally contains 23 pairs of chromosomes for a total of 46. Now, what it doesn't say, but I've added in here, is that the paired chromosomes should contain the same type of genetic information. And so for anyone who's a bit shaky on the biology they picked up in school, please think about those chromosomes like chapters in a book, containing lots of letters, which in a certain order will make up words, okay? So it's fair to think of sentences as genes. The genes or sentences are pretty important. So luckily we have a backup in case anything happens to the first one. And that's the issue for our 8p heroes. So one of their chromosome 8s no longer has 16 pages. We see that in orange here for caution. And thankfully, our backup book number 2 still has a chapter chromosome 8 in good working order. And I'm showing that in green for good here. Now, in this cartoon, we look at the 3 main variations of chromosome 8p disorders. And apologies to anyone who's watching this who has a 8p hero or 8q hero, which isn't included in this discussion. Now, the petite arm, the 8p arm, is denoted P, and below it, the Q arm. Now, the majority of 8p heroes will have one typical 8p chromosome 8 when typical 8p arm. But heroes with deletion 8p will have a section of DNA missing from the 8p arm. So that red DNA section is no longer part of the chromosome. And at some point, it's likely to get lost during the cell replication process and disappear from the cells. So chapter 8 in this book is missing a few paragraphs or sentences or pages, depending on the size of the deletion. Heroes with duplication 8p have had an extra section of DNA copied and inserted into the 8p arm. The blue section is a copy of the section below it. So chapter 8 in this book has a few extra sentences or paragraphs or pages which repeat themselves. And heroes with inverted, duplicated, deleted 8p, they'll have the deletion and the duplication, but their duplication will be facing the opposite direction and inserted inside, above or below the bit that is copied. It'll also have that deletion there. And if you wanted to read that, you would have to actually do it in a mirror because it's backwards. So what we need to understand is why genes are important. What do they actually do? Now, genes code for proteins. The normal process is happening here within a cell. You see chromosomes, as I've drawn them as scribbles, which are made of DNA. And the DNA contains lots of genes which code for messenger RNA, which I'm showing as lines. Now, the messenger RNA is translated into proteins, which fold up into shapes. And all these processes are happening within the cell. And I'm drawing the proteins as shapes which will interact with each other. And the way they are formed, the shape they take, dictates which shape they'll interact with. So going back to our book analogy, if the sentences in the book are genes, it is fair to think of the proteins as the characters in our story. They do all the actions. They move around and do things, interact, and cause chain reactions. Now, each organ in our body contains many cells, and the cells in each organ have different proteins. So cells all have the same DNA, but they will have different proteins present within them based on genes being on or off. This allows cells in different organs to look and function differently from each other. So you can see in this image, I've represented that by drawing proteins from the brain as arrows, stars, and Pac-Mans, from the heart as half circles, diamonds, and trapezoids, and from the stomach as timers, corners, and circles. Now, what do proteins do? They do everything. Proteins interact with one another to cause changes in a cell. So here I've drawn the proteins coded for both chromosome 8s in green, as this cartoon is showing us what it would look like if there were two typical chromosome 8s, i.e. no deletions, no duplications. So say in this scenario, having two normal chromosome 8s means having 6 green triangle proteins. And they come together to interact with this V-shaped black protein, which is coded for a gene on, let's say, chromosome 9. When they come together, they cause a change with an end result, which may make, for example, our muscles move or make us sleep or control our emotions or how our stomach processes food. Now, gene dosage is another important concept to understand. How many copies of a gene you have can affect how many copies of a protein and other cell regulatory factors are created. And this will have an effect on the cell, the organ, or the body. And let's think about these normal processes in the context of our 8p heroes. So heroes with deletion 8p are missing that chunk of chromosome. So their orange chromosome is unable to generate a triangle protein. So that means instead of having 6 triangle proteins, we only have 3. So maybe some of these processes will continue as normal, but some might not. Duplication 8p heroes have the opposite issue. They have some extra DNA, which is going to result in an extra circle protein. So again, consider the normal process where there's 2 typical chromosome 8s, again, no deletion nor duplication. In this scenario, 4 green circle proteins are what is required to make us feel the right amount of tired, to make us anxious, to control the tension in our muscles, to control our sense of balance. But now, in duplication 8p heroes, we may have too many circle proteins. And this might make them feel more tired or more anxious, too much tension in our muscle, and perhaps put our balance off. So again, some of these processes in our 8p heroes might be normal, but they might not. And what I put in here to just give everyone a sense of the scale of these things is a cartoon demonstrating just one of many, many protein signaling pathways which occur in human cells. So each of these colorful ovals is a different protein. You see that some proteins are connected to one another by arrows, and others are connected by T-shapes, which mean they inhibit change. So these proteins exist in different parts of the cell. You see some receptors are embedded up here in the cell membrane, and then others are in turn going into the nucleus to turn genes on and off. So the purpose of looking at this is just to give everyone an appreciation of how many different interactions may take place with all the different proteins within a cell. So we have looked at the normal process of DNA coding for messenger RNA, which is then translated into a protein. We understand that 8p deletions may result in less proteins, which may have an adverse effect in our 8p heroes. We understand that 8p duplications might result in more proteins, which may have an adverse effect in our 8p heroes. Now, our inverted duplicated deleted 8p heroes will have both the deletion and the duplication, but their duplications are inverted, which means they are running in the opposite direction to the DNA in the 8p arm that they are inserted into. So this is likely to cause more disruption to the genes in the vicinity at either end where they are inserted. And and it's difficult to quantify, and is going to be different for every single 8P hero. So for this reason, I've just sort of put question marks here as to the effect of that for our 8P heroes. Now, there's one final factor to also consider when thinking about 8P heroes and their medical presentation. And here we're looking at a cartoon of those first steps of embryogenesis, when the fertilized egg starts to replicate and divide. So this is when one cell becomes two cells become four, et cetera, et cetera. So if the 8P chromosomal anomaly happens here at fertilization, all cells within the 8P hero will likely contain the same 8P anomaly. But if the chromosomal anomaly happens later, during embryogenesis, then it is possible that only some of the cells contain the 8P genetic anomaly. So here, for example, I've indicated the genetic mishap occurring when baby is only 8 cells big. And you can see how, as those cells replicate, they're only going to be in a proportion of the body, not the whole body. And this is called mosaicism. Now, everything I've said so far was understood in 2012. And that is when I first came to learn about these conditions, when my daughter was diagnosed with inverted duplicated deleted 8P. So at that time, we were told the genetic anomaly is very rare and the outcome variable amongst those diagnosed. We can't predict how your daughter will develop, but she will not develop at the same rate as her peers, and she will never live independently. Now, there were no major studies into the similarity of children with variable 8P chromosomal anomalies at this time. And I had done a PhD in cancer biology, and I'd done postdoctoral work in stem cell research. And I thought the potential information that could come from studying all these genetic variations was extraordinary. But when I first started to see if I could generate any scientific interest in 2016, no one I spoke with was interested. They thought it was going to be too complicated. It would be impossible to gather enough information from participants to put a meaningful study together. Now, in 2012, my daughter had a spell of really bad health. We now think that she had actually developed cyclic vomiting syndrome. So it was during that time, in absolute desperation, that I sort of returned to Google and asked it about, inverted duplicated deleted 8P. And much to my absolute surprise, I stumbled across Project 8P, and I really couldn't believe my eyes.